Hello everyone, I am Dr. Kavita Gajpati, Assistant Professor in Pune College of Pharmacy, Bharatiya Vidyapeeth, deemed to be University. So today's topic for presentation is viruses. So the contents are introduction, is structure and morphological characteristic of viruses, life cycle, or we can say the replication or multiplication cycle of viruses. It is classified into two categories, lytic and lysogenic. And then the transmission process of viruses and different kind of infections, and also the antiviral drugs. So first of all, we will start the introduction and the definition of viruses. What are the viruses? Viruses are actually submicroscopic entity consisting of a single nucleic acid surrounded by a protein coat and capable of replication only within the living cells of bacteria, animals, or plants. So that's why these are classified into three broad categories. For example, when these viruses use bacteria as a host, so these kind of viruses are known as Phasenes. When these lives on the animal host, so these kind of viruses are known as Zoophasenes. And when these lives on the plant, or these viruses are used plant as a host material, so that time the type of viruses are known as phytophagenes. So we can say these are the obligate intracellular parasites. Now, in another way, we can say these are the submicroscopic infectious agents that replicate only inside the living cells of an organism. And viruses infect all life forms of human beings and also plants and also different kind of microorganisms. And it is an infectious microbe consisting of a segment of nucleic acid, either DNA or RNA, surrounded by a protein coat. Sometimes it has not been surrounded by the protein coat, or sometimes it has surrounded by the protein coat. So, on the basis of surrounding, these are also again classified, or we can say the enveloped and non-enveloped viruses. And these viruses cannot replicate alone, and it must infect cells and use component of the host cell to make copies of itself. That's why these are known as the obligate intracellular parasite. So this is the simple virion structure, matured virus structure. Here you can see in the internal portion, virion associated polymerase are present, and the nucleic acid, it is also present in a circular form. And these nucleic acid or genetic material surrounded by the protein capsid. And these capsid prepared by the small monomer units, which is known as capsomere, and form a proper coating which coat or protect the nucleic acid. And surrounding that capsid, one more envelope is present, which is prepared by the lipid or lipid envelope, we can say. And over that lipid envelope, few kind of projections are present, which is known as spikes or some kind of attachment moieties. And by these spikes or projections, viruses can easily attach with the host cell, and that attachment, or we can say it can easily release their genetic material and penetrate in that genetic material inside the host cell easily. And if we talk about the morphological characteristic or morphology of viruses, so these are broadly classified into three categories. For example, simple structure. In case of simple structure, we can say the helical structure of virus that we will discuss in further slides. And repetitive structure, here also we can say enveloped helical structure, means helical structure which is covered or protected by some kind of envelope. And high level of redundancy, for example, bacteriophage. So here you can see the different structure of viruses, and For example, if we say about the helical viruses, so helical viruses contain capsid, and also it has RNA as a genetic moiety. And for example, we can say the tuberculous viruses, which infect a normal of different type of number of different types of plant, have a slinky shaped capsid that twists around and encloses its genetic material. And if we take the polyhedral viruses, so here the example of this category is adenoviruses, which are known to cause a range of illnesses from pink eyes to pneumonia, and are composed of genetic material surrounded by a many-sided capsid, usually with 20 triangular faces. And the third category is spherical viruses. So these categories are classified on the basis of some kind of shapes of viruses. So here, in case of spherical viruses, we can say our well-known coronavirus and are the essentially helical virus enclosed in a membrane known as an envelope, which is spiked with surgery protein and assist in sticking to and entering host cell. And last one is complex viruses and viruses having more redundancy. So these kind of viruses generally infect the bacteria. That's why this is known as bacteriophage, which infect or kill the bacteria, resemble a lunar lander, and are composed of a polyhedral head and a helical body, tail, sheath, and legs, or we can say the tail fibrous portion which is present, and that attached to a cell membrane so that it can transfer its genetic material easily. So you can see here in polyhedral viruses, DNA is present as a genetic material, capsid covering is present, and glycoproteins covering is also present. Similarly, in case of coronavirus, spherical viruses, capsid material, RNA as a genetic material, and glycoprotein covering with spikes. And here also, complex virus bacteriophage, you can see some different kind of structure. DNA, head, and tail, sheath, and that fibrous portion of tail, which is capable or which is useful for attachment, proper attachment with the host cell. Now I am coming to the point virus replication. So here specific 8 steps are present. In this manner, virus can replicate or multiply within the host cell. So first of all, virus attachment and entry point. First point or first step is attachment and entry. Second point, you can see here, uncoating of virions. Uncoating of virions. And then third point is migration of genome nucleic acid to nucleus. Yes, that nucleic acid to nucleus migration occurs in the third step. Fourth step is the transcription process. Fifth is the replication process. And sixth is translation. After transcription, that we can say DNA to mRNA formation and translation of viruses into mRNA and virion assembly. After that, virion can again assemble and release of the new protein viruses particles. So you can see here in the 8th step. So on the basis of this replication, we can classify the virus life cycle into 2 specific classes, lytic cycle and lysogenic cycle. So what is actually the lytic cycle? It is more common and a method of viral multiplication wherein the virus attack a host cell. It destroys the host cell totally by feeding on the metabolism of the host in order to multiply. And just opposite lysogenic cycle, it is not a common method of viral reproduction or multiplication. Majority is dependent on the lytic cycle only. And in this lysogenic cycle method, the virus unites its genetic details with that of the host, turning dormant and lets the host to reproduce while continuing its regular activities. Although similar at times, understanding the difference between the lytic and lysogenic cycle is very important. So we will just discuss the differences between these two cycles also. So here lytic cycle, if we talk about this, so the DNA of the virus does not integrate into the host cell, but in case of lysogenic, DNA of the virus integrate into the host cell. And lytic host DNA, hydrolyzed host DNA does not hydrolyzed in case of lysogenic. Absence of prophase stage, but here lysogenic presence of prophase stage. Here, lytic cycle DNA replication of virus takes place independently from the host DNA replication. But here, just opposite in case of lysogenic, DNA replication of the virus takes place along with the host DNA replication. And lytic cycles occurs within a short period of time, and lysogenic takes time. Lytic cycle symptoms of viral replication are evident. Here, lysogenic symptoms of viral replication are not evident. Genetic recombination in the host bacterium not allowed, but in case of lysogenic, it is allowed. Lytic cycle, the cellular mechanism of the host cell is totally undertaken by the viral genome, but here it is the mechanism of the host cell is somewhat disturbed by the viral genome. So these are some kind of differentiative points between both kind of cycles. So here you can see both type of cycle, that diagrammatic presentation. So this is the bacteriophage. You can see here in the first point in lytic cycle, bacteriophage attach and release its genetic material and phase attached to the host cell and inject DNA. DNA comes inside the host cell. And these, in the second point, this DNA or the host cell genetic material circularize and entries lytic cycle. And now here the biosynthesis, or we can say generation of a different component of bacteriophage. Biosynthesis of different component of bacteriophage occurs in this lytic condition in third step. And after that, if we see the cell lysis release phase virions. Virions are the mature bacteriophage, and after maturation inside the host cell, it can easily release by bursting reaction. So this is the lytic cycle. And if we talk about the lysogenic cycle, so after entry of DNA or penetration of DNA, that host cell genetic material and viral genetic material can integrate with each other. Here you can see easily in the third step of lysogenic cycle. So phase DNA integrate with the bacterial chromosome, and then this integration makes possible to prepare or reproduce some kind of bacterium or clone's body. So lysogenic bacterium reproduce normally. Here you can see the several cell division occurs and then the formation of two different entity, or occasionally the prophase excises from the bacterial chromosome and enters the lytic cycle. So here you can see the formation of different two clones body, and again if the type of cell is remain, it enters into the lytic cycle also. So this is the kind of lysogenic and lytic cycle. Now coming to the point, transmission of viruses. There are lots of ways by which viruses can easily enter inside the human body. So it may be the respiratory transmission, for example, influenza A virus. It may be the fecal oral transmission, for example, enteroviruses. Blood-borne transmission may occur, for example, hepatitis B viruses. Sexual transmission also occurs, for example, HIV viruses and human immunodeficiency viruses, and animal or insect vectors, rabies viruses. Few in vivo disease process also occurs in case of viral transmission. And after viral transmission, it causes cell destruction, deterioration, some kind of biological changes of the cell, some kind of mutation, alteration may also occur so due to the viral infection. And virus-induced changes to gene expression also. Yes, that's why this is the cause of mutation. And immunopathogenic disease also developed by the viruses. So if we take the point about the cancer or human tumor condition, so here we can say Epstein-Barr viruses are responsible for Burkitt's lymphomas. Human papilloma viruses are responsible for benign warts and cervical carcinomas in the human body. Human T-cell leukemias viruses cause the leukemias. Hepatitis C viruses are the main reason for liver carcinomas. So there are lots of transmission ways or lots of disease or basically cancer is very dangerous disease, or if we are talking about this, so we have already taken few examples related to that. So viruses are also responsible for generation of the type of mutation, alteration, gene expression, deformation of cells, and cause the tumor-related disorder or diseases in our human body. Yes, so how it induced a tumor inside the body? So viral infection, we can say it is responsible for the generation of mutation, expression changes, and uncontrolled cell growth and tumor formation occurs due to the viral infection. So this slide represents how virus infect and how it is responsible for multiplication, replication, and production of more clone copies inside the human body, either the lysogenic or lytic cycle. So virus in this series, we can say virus infects the cell, virus nucleic acid DNA integrate into cellular genome, and virus cause changes into the cellular gene expression and form the uncontrolled and cell manipulated cell multiplication and tumor formation, or lots of a number of uncontrollable clone bodies formed by the viruses. So you can see this is the normal cell and this is the transformed cell. Normal cells expressed or represent as some kind of flat form or spreaded form, but here in the transformed cells you can see some kind of nodules and puffy patches are present, which represent as a defective part of the normal cells. Okay, so here overviews of some viral infections. So we can say encephalitis, meningitis due to some JC viruses, rabies, arboviruses, LCM viruses, common cold due to rhinoviruses and para-influenza viruses. Lots of viruses are here. Eye infection generally by the adenoviruses, herpes simplex viruses, parotitis, mumps virus responsible for that, and gingivostomatitis, herpes simplex viruses, pharyngitis, adenoviruses, cardiovascular diseases, Coxsackie B viruses, hepatitis viruses, skin infection, varicella, human herpes viruses, smallpox, human papilloma viruses, rubella, measles, sexually transmitted disease, for example, here HIV, human papilloma viruses, herpes simplex type 2 viruses. So there are lots of diseases in human body. So viruses are responsible for generation of the type of diseases inside the human body or taking as a host of human body. So some antiviral drugs, which is very famous and which target specific areas inside the body, like or which kind of viruses are responsible, like herpes viruses. So here, in case of this, we generally use vidarabinine, and which target the virus polymerase, herpes simplex, acyclovir, which is very popular drug, and it also target virus polymerase, and cytomegalovirus gencyclovir is very useful as an antiviral drug. So this also target virus polymerase, and similarly retroviruses, squenavir, ritonavir, few of the antiviral drugs and which target HIV protease. So lots of drugs are also available in the market as a brand or as a very famous or popular antiviral preparations which treat viral diseases easily or in very efficient, effective manner. Thank you so much.